In this video, we're going to continue with looking at common issues with Punnett squares, and this time I'm going to show you some shortcuts. Shortcuts that will make doing the Punnett squares a little bit easier, some patterns that we can look for. And the reason we want to do these shortcuts, not because we're lazy, although I am, but mostly it's because Punnett squares are complicated, they're big, there's lots of little letters in there, and they're prone to mistakes. So any way you can simplify a Punnett square and lead you to an answer more quickly and more efficiently, it's a good thing. So let's look at the example we had in a previous video about tomato plants with purple stems dominant over green stems. So big P is equal to purple, and little p is equal to green, and that's their stem color. And then it says that tomatoes are either red, dominant over yellow. And that's the color of the tomato itself. All right, it also tells us these genes are located on separate chromosomes, which means they follow the law of independent assortment, which means a Punnett square is an appropriate way to go about solving a genetics problem. So let's take a look at a cross of the parent generation. Let's say that in our P generation, we have a purple plant that is homozygous purple, two big P's, and let's say it's heterozygous for being red. Big P, big T, little t. And let's say that we cross that with an individual that is, let's say it has green stems and also heterozygous for red tomatoes. Now, if we want to do this cross and find out the F1 generation, we would set up our Punnett square by doing FOIL. First, outside, inside, last. We covered that in a previous video. First P with first T, outside P with outside T, inside P with inside T, and last P with last T. And if we were to do that, we would end up with a big P with a big T, a big P with a little t, a big P with a big T, and a big P with a little t. And doing the same thing for the other parent, we go first P with first T, outside P with outside T, inside P with inside T, and last P with last T. And we would end up with little p big T, little p little t, little p big T, little p little t. And we can fill out a Punnett square for that. All right, and I'm going to pause the video while I fill out the Punnett square. We'll be right back. All right, we're back with the completed Punnett square, and we can now find out our genotype-phenotype ratios. Let's go with phenotype ratio for this one. Of course, that depends on what you're being asked in a particular question. But our phenotype ratios here, we can see that we have right here purple and red. So that's purple stems, red tomatoes. And we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 of those. So 12 purple stem red tomato. And then we also have, if we look right here, we've got purple stem yellow tomato. So we have 1, 2, 3, 4 of those, and that's our whole Punnett square. We have a 12:4 ratio, which of course can be reduced into a 3:1 ratio of purple red to purple yellow. But what if there is a quicker way to figure this out? And of course there is. So what I'm going to do is I'm going to erase the Punnett square that I have here, and we're going to take a closer look at the gametes that we got from the parents by doing FOIL. So let's just erase all of this right here. And let's see if we can't gain some efficiencies here, figure this out a much quicker, less involved way. Now, the shortcut I'm going to use is called eliminating redundant gametes. A redundant gamete means a gamete that has been listed once already in our Punnett square. For example, if we look at this parent right here, we have a big P and a big T, and then I see the same gamete again. I'm going to eliminate that. I'm just going to cross it off. I've already got it once. I don't need it twice. Because what that means is anything I get in this row is going to be exactly the same as anything I get in that column, I guess, in the row. So it's going to be the same. So I don't need to do it twice necessarily. And I can see another redundant gamete. I have a big P, I had a big P, little t here, and I have another one here, which means I can eliminate that one. I can do the same thing going down the side of my Punnett square. I've got a second little p big T here, which matches up with this one, and I have a little p little t right here that's redundant. So if I eliminate those redundant gametes, just get rid of them, let's see what happens to my Punnett square. The first thing is it's going to be a much smaller Punnett square. It's going to be a Punnett square of only 4 squares instead of 16. So right there, it's going to save me a fair bit of time filling it out. That also means I have fewer opportunities to make silly mistakes that people are prone to making, myself included, as I've shown in my previous videos using Punnett squares. I end up doing a fair bit of erasing and rewriting. All right, so what we end up with here is a 4 square Punnett square. But when we take a look at the result, we get the same result we got before. Purple and red, we now have 1, 2, 3. In our previous Punnett square, we had 12. Well, here we have 3, which is what it reduced to anyway in our ratio. And we have 1 purple stem yellow tomato. Well, we have 4 in our previous Punnett square that we reduced down to 1, which means we get a 3-to-1 ratio, regardless of whether we do a full Punnett square or a shortened Punnett square. So, eliminating redundant gametes is a good habit to get into for saving a lot of time and making a Punnett square and reducing the chance for making errors when writing out that Punnett square. So that's the first shortcut that I've shown you. Let's look at another shortcut. I'm going to take a different P generation. And this time I'm going to show you a shortcut that only works in one circumstance. It's a very specific shortcut. And that circumstance is if we have both parents heterozygous for both traits. In other words, both parents are big P, big T, little t. In a situation like that, we do FOIL to set up our Punnett square to figure out the offspring here. We would get big P, big T, big P, little t, little p, big T, little p, little t. There are no redundant gametes here. Okay, I don't see any of them twice. They're all unique. Now we have the same thing down the side. Because it's the same genotype, we'll get exactly the same gametes. And we end up with a 16 square Punnett square. We cannot use the previous shortcut in this case because there are no redundant gametes, so I can't make this down into a 4 square Punnett square. I have to write out all 16 of them. So once again, I'll pause the video and I'll be back when it's all filled out. So here we are back with the Punnett square all filled out. Now if I want to write the phenotypic ratio for this, I have 4 possible outcomes here for phenotypes. We've got a certain number of purple stem red tomatoes. We've got a certain number of yellow stem red tomatoes. We've got a certain number of purple stem yellow tomato, and we have a certain number of, sorry, this was green here. Start that again, let's make that green. And we've got a certain number of green stem and yellow tomato. All right, so we have 4 possible different phenotypes that we could get in this Punnett square. So if we figure out the number of purple and red, 1, 2, 3, 4, 5, 6, 7, 8, 9 that are purple and red. And then green and red. Green and red would be 2 little p's and at least 1 big T. 1, 2, 3 of those. And then purple and yellow. Purple and yellow would be one big T and two little t's, or at least one big T. So we've got 1, 2, 3 of those. And that leaves us only one in our Punnett square, little p, two little p's, two little t's, which would be green and yellow. So we get 9 to 3 to 3 to 1. Now, there is a shortcut we can do here. Because we can't use the other shortcut of eliminating redundant gametes, what we can do here is, I'm just going to change things up a little bit. I'm going to take away this specific phenotypes we have here. And I'm just going to write it this way. We're going to end up with 9 that are dominant for the first trait and recessive for the second. Sorry, dominant for both traits. Let's get that right. Dominant for both traits. In other words, purple and red, 9 of those. Then we're going to have 3 that are recessive for the first trait and dominant for the second trait. So recessive for the first trait means green, and dominant for the second trait means red. So green and red. Then we're going to get 3 that are dominant for the first trait and recessive for the second trait. That means purple stems, dominant for the first trait, and yellow tomatoes, recessive for the second trait. And then lastly, we're going to get 1 that is recessive for both traits. So that means green stems and yellow tomatoes. And when we have parents that are heterozygous for both traits, and it's both parents heterozygous for both traits, we will always get a 9 to 3 to 3 to 1 ratio of 9 dominant for both traits to 3 recessive for the first, dominant for the second, to 3 dominant for the first, recessive for the second, to 1 recessive for both traits. So anytime you have, and it only works in this situation, heterozygous for both traits times heterozygous for both traits, then there's no need to even write a Punnett square. We can go straight to 9 to 3 to 3 to 1 ratio. It will be that every single time. Cautionary note on this, this works great when you have a simple dominant and recessive scenarios. If there's anything to deal with X-linked traits or codominant traits or traits that interfere with one another, we'll do another video about that a little bit later. Anything that's not just standard dominant recessive, don't use the shortcut. The shortcut has very few applications, but it can save you time in writing a whole Punnett square. So let's take a look at a couple of exam questions, and we'll see just how quick they are to solve when you do the shortcuts. So this first one deals with p's. The allele for tall, big T is dominant over the allele for short. So big T equals tall. I always write out what my symbols mean. Helps me keep on track and what I'm talking about here. So big T tall, little t short. And then we have axial flower position A over terminal flower position little a. All right, then we are told that the allele for plant height sorts independently. We always want to see that. That means they're on separate chromosomes. Sometimes it'll say these alleles are on separate chromosomes. Sometimes it'll say they sort independently. That just means a Punnett square would be an appropriate way to solve the problem. So a plant heterozygous for both traits. Or even read the rest. I'm going to write that out. Heterozygous for both traits. Big T, little t, big A, little a. Is crossed with the plant homozygous recessive for both traits. So homozygous recessive for both traits, little t, little t, little a, little a. What percentage of the offspring produced would be expected to display at least one of the dominant traits? Well, to get that, we need a Punnett square. So we do FOIL in the first parent, and we will get big T, big A for the first gamete, big T, little t, big A, and little t, little a. Now, for the second parent, we're going to get little t, little a, and when we do FOIL, they're all going to be the same. They're all going to come out little t, little a, little t, little a, little t, little a. So what we learned earlier is we eliminate redundant gametes. Just get rid of those. Don't need them. Which means our 16 square Punnett square is now a manageable 4 square Punnett square. So big T, little t, big A, little a, big T, little a, little a, little t, big A, little a, and little t, little a, little a. Now the question is asking us for the percentage of offspring expected to display at least one dominant trait. So at least one dominant trait could be both dominant traits. So we have one that has two dominant traits, this one has one dominant trait, this one has one dominant trait, and this one does not, which means we have 3 out of 4, and our answer is 75%. Much quicker way to get the question done when we have a manageable Punnett square. All right, the next one here deals with coat color in dogs. This one's a little bit more complicated because we have two traits that interfere with one another. The dominant allele big E produces a black coat. The recessive allele little e produces a red coat. However, how that gets expressed, well, there's some more information here. It says another gene controls the expression of color. The merle alleles are incompletely dominant as shown below. So we've got little m is equal to full color, big M little m is equal to dilute color, which could produce a gray or a light red, and big M would be white, so completely white dog. So in this question it says a gray dog that is homozygous dominant for the eumelamin mates with a dark red dog. So a gray dog, we've got gray right here, that means they must be big M little m, and it says that it's homozygous dominant for E's. So that means our first dog in our P generation is big E, and then it must be heterozygous to come out gray. It's not white, it's not black, it's gray. All right, and that is crossed with a dark red dog. So a dark red dog, dark red is right here, must have little m, and to be red, it must have two of the recessive small e alleles. So we've got this situation. And in this case, we can make quite a compact Punnett square, because when we do FOIL and we eliminate redundant gametes, for first parent here, we could get big E with big M, or we could get big E with little m. Doing FOIL, we'd end up with 2 of those, and we'd end up with 2 of those. We can eliminate the extras and just write down the 2 of them. For the other parent, there's only one possibility. When we do first, outside, inside, last, we do the FOIL, they all are going to end up as little e, little m, which means we have a 2-square Punnett square. Big E, big M, little m, big E, little m, little m. Much, much shorter than doing a 16-square Punnett square, much more manageable, and the answer comes out right off the bat. We can see that here we're going to have a big E for the black color, but it's only going to be diluted, so that means we're going to have gray. And here we're going to have the black color, however with the little m, that's going to end up being full color, okay, full color up here, which means this will be black, which means we will end up with half of the offspring gray. We'd expect half of the offspring gray and half of the offspring black, and we would pick answer A.